I'm very happy to be with you right now. So, dear participants, on behalf of the organization committee, it is my great honor and pleasure to welcome you to the 8th Ankara-Hannover Cooperation Symposium. This is my second moderator job to a joint Hannover event, which I'm very happy and excited with right now, where the first one was previously held in June on digital technologies in veterinary education and animal health. I would like to thank Dr. Lepenies for realizing in this event in the Hannover part and Hannover Commission in Ankara University. Our symposium will start with the opening speeches by the Dean of Ankara University Faculty of Veterinary Medicine, Professor Dr. Ender Yarsan, and continue with the President of the University of Veterinary Medicine in Hannover, Dr. Gerhard Greif. We will then continue with the Joint Relations Coordinator of both universities, Professor Bernd Lepenies and Professor Hakan Öztürk. The opening part will be ended by the presentations of Dr. Manfred Kietzmann and Dr. Mehmet Şahal on the memories of the joint collaboration. Following the opening speeches, we will continue with the scientific session on infectious diseases, starting with Professor Aykut Özkul. Professor Özkul is the head of the Department of Virology and the Institute of Biotechnology in Ankara University, who is also a member of the Coronavirus Scientific Advisory Board in Turkey. His presentation is Diversity of Zoonotic Arboviruses in Turkey. We'll then continue with Professor Paul Becker, the Director of the Institute of Virology in Hannover and Head of the Specialist Group Virology and Virus Diseases of the German Veterinary Medical Society, with a speech on virus research at the Institute of Virology, TIHO, selected examples. Following Professor Becker's presentation, we'll be following Professor T.Çiğdem Oğuzoğlu from the Department of Virology in Ankara University, who has completed her PhD in Hannover with a special interest in molecular virology and vaccine development. Her presentation will be expression of protein baculovirus system as a vaccine candidate, which is currently a very hot topic. We will then continue with the COVID again by the presentation of Professor Asisa Volz. Professor Volz will be presenting immunogenicity and efficacy of COVID-19 candidate vector vaccine MOA SARS-2S in preclinical vaccination. Following Professor Volz, we'll be following Professor Valentin-Weigand, the head of the Institute of Microbiology in Hannover, with research activities focused on molecular mechanisms of pathogenicity in bacterial infections, vaccine research, and in molecular epidemiology. The session will be closed by the presentations of the parasitology experts. Dr. Gökben Özbakış Beceriklisoy from the Department of Parasitology in Ankara will give her speech on a retrospective review on zoonotic toxicity in Turkey and Germany. Her research areas include molecular parasitology with a special focus on zoonotic helminths. Professor Christina Strube from Institute of Parasitology in Hannover, with research studies in her laboratory, including epidemiology of parasitic infections in farm animals, domestic animals, and wild animals, with focus on helminths and arthropods, will be presenting a neglected zoonotic parasitosis in neglected people. The meeting will be held from this Blackboard system. For the presenters, a quick reminder to allow pop-ups for the screen sharing. The scientific sessions will be held for 10 minutes presentation with 5 minutes QA session. If you have any questions and comments, you can write on the chat session and or raise hands following the presentations. If the time exceeds, I would kindly ask your questions directly through contacting the presenter from their mails. I would like to remind you that participation certificate will be issued to participants who are registered. I also would like to remind you that keep your microphones closed throughout the session. The time between Hannover and Ankara is 2 hours, which has recently changed this weekend, and thanks to the understanding of our Hannover partners, we are continuing off the schedule 1 hour earlier than expected for the German side, and hope that we'll meet the presenters on time. In case of delays and technical difficulties, we may be shifting the other presentations and would like to apologize beforehand. Thank you very much for your kind contribution and wish to have a fruitful collaboration symposium. Okay, so thank you very much. And now I leave my word to Professor Dr. Ender Yarsan, the Dean of Ankara University.
Thank you very much, Begüm. Good morning to everybody. Dear President of the Veterinary University of Hannover, dear ladies and gentlemen, dear students, and dear participants, I am very happy to greet you at the Ankara-Hannover Cooperation Symposium, which we held for the 8th time today. As we all know, the most basic missions of universities are education, research, and contribution to society. Universities always aim to improve quality when performing these basic missions. In this context, national and international cooperation are of great importance. The cooperation agreement between Hannover University, Hannover Veteriner University, and Ankara University Veterinary Faculty has provided great gains to our institution in the past, and I hope that this agreement will make significant contributions to our institutions in the future. Here, I would like to underline that the cooperation agreement between our universities is very special and has historical foundations. The cooperation agreement between our universities, unlike the classical protocols, this agreement that was published in the official Gazette of the Republic of Turkey on January 7, 1981. However, the records of the relations between Germany and Turkey in the field of veterinary medicine date back. Russian Prince August sent military veterinarian Godlewski to Istanbul in 1843 to support the Ottoman Empire army. Godlewski founded the military veterinary school in Istanbul in the same year. This school was moved to Ankara in 1933 and formed the basis of Ankara University, Faculty of Veterinary Medicine. In this process, many German professors served as deans in Ankara and worked as faculty members in different departments. As part of the cooperation between our universities, many German scientists came to Ankara from Hanover and conduct research. Every year, Turkish students from Ankara complete their summer training within the scope of cooperation agreement, and many find an opportunity through Erasmus placement and mobility programs. Many researchers and students from Ankara visit Hannover Veterinary University in this context as well. In almost every department of our faculty, there is a faculty member with a doctorate degree or have a direct contact with Hannover. Dear participants, I am very pleased to see you among us at the 8th Cooperation Symposium. I hope it will be very successful. I respectfully greet you all. Thank you very much.
Thank you very much, Professor Yarsan. And Dr. Greif.
Thank you for the introduction and opening the session, dear Professor Yarsan, dear colleagues. It is my great pleasure to be present at the beginning of this new format of collaboration between our both renowned universities. So good morning to everyone, to the students, to the teachers, and to everyone who has been involved in the past and who will be involved in the future in this collaboration between Ankara University and TO in Hanover. Special thanks goes to Lepenies and Hakan Öztürk. I guess they were the ones who really brought it forward, but many others who contributed a lot and will do so in the course of the day. So cooperation, as already mentioned, is historic, has a historic dimension, and I will not touch on it because we will hear more of it by the colleagues which are to come in the next couple of minutes. It has been mentioned, why are universities there? Universities are there for education, first and foremost. That is true. But education which is based on research, and this is very important, that research makes a university from a school really to a university. And something which is brought about by that, this is innovation. Innovations which are brought to society, and let me also mention one thing should be added, and this is a form of a cultural exchange, especially in international collaborations. What it's all about is about people, and people work together, people collaborate, not institutions. So I really hope that we find a way that we have more and more people who are interested in this collaboration again, and that is my hope for the future. Identification of areas of mutual interest, of mutual benefit for the good of our societies. Get the people together in research projects, in educational projects, PhD programs, for example. Seeks the funding for it, ideally on a European basis, is very important because there is the most money, more than in our universities. So go for that one and jointly. Let me say one thing, our relationship, not only our mutual relationship, but also the relationship between all universities and the societies in Turkey and Germany has been interrupted recently within our countries and between our countries. COVID-19 is a name, so we will hear more about it in the course of the day, and this is to confirm that research in the field which was of our mutual interest for a couple of decades and will be so in the future. Infectious diseases research is so important for our societies, for our universities, for all of us. Now, time is running. I'm looking forward to regain in momentum for this collaboration. The broader the personal exchange, the better. The long-term perspective will be based on these mutual interests and on the individuals, on the people, on the young people which we educate and exchange in the next couple of years. So I'm looking forward to an interesting day, and thank you for allowing me a few words.
Thank you, Dr. Gerhard Greif. And now is for Dr. Bernd Lepenies.
Yeah, dear Professor Yazan, dear Dr. Greif, dear colleagues, dear students, dear friends, it was a pleasure having participated in the co-organization of this conference, of this symposium. I'm really looking forward to it. I just took over the responsibility here as being the responsible for the partnership between both universities last year, and it's really a pleasure. And we have already heard that this relationship is historical. And what we'll hear during this conference is that there are long-standing collaborations that have been very productive and successful, and there will be more to come. This is the idea of this conference, to, as Dr. Greif already mentioned, identify areas of mutual interest and continue collaborations being successful here. I just joined the University of Veterinary Medicine about 6 years ago, so working in the field of infection biology. So didn't have the time to visit Ankara yet, but we'll hear examples, we'll hear stories, memories from Ankara visits and visits also in Hanover during the conference. I'm looking really forward to my first visit to Ankara, and I plan to do so in the near future. Latest post-corona, we have heard that COVID-19 has been here, of course, a limitation, but this will be overcome. And this conference may be a starting point here, so I'm really looking forward to this conference. And not to take any time from the clock, I'll hand over to Professor Öztürk here. Thank you.
Thank you very much, Bernd. And now we're leaving to Professor Hakan Öztürk.
Dankeschön, Begüm. Sehr geehrter Herr Präsident, sehr geehrter Herr Dekan, meine sehr geehrten Damen und Herren, liebe Studentinnen und Studenten, ich freue mich, Sie bei dem gemeinsamen 8. Kooperationssymposium der DSGN Fakultät der Universität Ankara und der Stiftung der DSGN Hochschule Hannover begrüßen zu dürfen. Es ist auch Herr Dekan Professor Yazan dargelegt hat, haben die Beziehungen zwischen Deutschland und der Türkei auf dem Gebiet der Veterinärmedizin eine langjährige Vergangenheit. In diesem Jahr begegnen wir das 40. Jahr der offiziellen Zusammenarbeit zwischen unseren Institutionen. Dank dieser Kooperation war auch ich unter der Betreuung von Professor Gerhard Breves, Doktorand des Physiologischen Institutes der TSN Hochschule Hannover. Ich freue mich sehr, heute meinen sehr verehrten Doktorvater digital begrüßen zu können. Nach meiner Promotion in Hannover 2003 hatte ich bei der Abschiedsparty im Physiologischen Institut gesagt, dass ich eine Brücke zwischen Hannover und Ankara sein wolle und mein Bestes tun werde, um die Beziehungen zwischen unseren Universitäten zu verbessern. Das war mir stets ein besonderes Anliegen. Auch jetzt in dieser Funktion liegt es mir am Herzen. Ich hatte die Freude, viele gemeinsame Publikationen mit Herrn Breves zu veröffentlichen. Ganz besondere Freude hat es mir bereitet, das Buch Physiologie der Haustiere von Herrn Breves ins Türkische übersetzen zu können. Jetzt ist dieses Buch eine wichtige Quelle für Studenten der veterinärmedizinischen Fakultäten in der Türkei und auf Zypern. An dieser Stelle möchte ich zwei für mich prägende Erinnerungen in Verbindung mit Herrn Greif erwähnen. Die erste ist, dass er anlässlich seines Besuches im Institut für Physiologie in Hannover von meinem Geburtstag erfuhr und mir zu meinem Geburtstag persönlich gratulierte. Es hatte mich sehr beeindruckt, dass er als Präsident mitgefeiert hat. Meine zweite unvergessliche Erinnerung ist, dass er mich ungeachtet seiner Funktion als Präsident nach unserer Teilnahme an einem Kongress in Göttingen zur gemeinsamen Rückfahrt mit seinem Dienstwagen nach Hannover eingeladen hatte. Sehr geehrte Damen und Herren, ich wünsche allen Teilnehmern einen erfolgreichen Verlauf der Veranstaltung, viel Spaß sowie allen Anwesenden eine produktive und gute Zusammenarbeit. Herzlichen Dank für Ihre Aufmerksamkeit.
Okay. Thank you very much, Professor Hakan Öztürk and Professor Gerhard Breves.
Well, good morning, ladies and gentlemen, dear colleagues. First of all, I would like to thank the organizers of this symposium to invite me to talk about some personal memories of the Ankara-Hannover Cooperation. In addition to a number of successful cooperations between Ankara and Hanover, which have already been mentioned briefly, I would like to mention some of my own experiences. In 1999, I have visited Ankara for the first time, and I had the opportunity to see most of the departments of your faculty. And in the Department of Physiology, I met for the first time in 1999 Hakan Öztürk, who was a young graduate student at that time. He applied to do his thesis in my group, and we managed that he could come to Hanover in autumn 1999. He started to work on a project which aimed at characterizing potential effects of yeast cells on rumen microbial metabolism. He did these studies under in vitro conditions by applying a semi-continuous rumen simulation technique, the so-called rumen simulation technique, or briefly RUSITEC. He could demonstrate that yeast cells are a substantial substrate for microbial fermentation rather than a probiotic organism to stimulate microbial fermentation and growth. Hakan finished his thesis in 2003, and the quality of his project is underlined by the fact that he could publish his results in the Journal of Dairy Science, which is one of the most reputed scientific journals in ruminant physiology. When he left Hanover, we could provide him with a RUSITEC system, which has enabled him to continue this kind of research in Ankara. And as far as I have known from him, he is quite successful in that range. The close cooperation could not only be continued in certain research areas. On the occasion of one of Hakan's further visits to Hanover, I gave him the new edition of our textbook on physiology of domestic animals, and Hakan simply mentioned, "I will translate this book in Turkish." To be honest, I definitely thought that this would never happen. But to my big surprise, I have received this textbook in Turkish 2 years ago, translated and edited by Hakan Öztürk. The only thing I can say is congratulations. Taking together these experiences, it has to be concluded that these are good examples of a successful cooperation with regard to both research and teaching. In 2004, I have visited Ankara again together with some colleagues from Hanover, and as in 1999, we have highly appreciated the cordial reception and the great hospitality at the Ankara Faculty of Veterinary Medicine. I wish you all the best to further develop and intensify this successful cooperation between Ankara and Hanover. Thank you very much for your attention.
Thank you very much, Professor Breves, for your presentation. And coming to Professor Mehmet Şahal, please. Hocam ekran sunum paylaşımı yapabilirsiniz.
Tamam. Guten Morgen zusammen. Ich begrüße Sie alle Teilnehmer aus Hannover und Ankara ganz herzlich willkommen zum 8. Wissenschaftlichen Symposium. Ich freue mich, dass wir miteinander haben. Ich bedanke mich auch das Organisationskomitee für Ihre Bemühungen ganz herzlich. Die Beziehungen zwischen der Tierärztlichen Hochschule Hannover und der Fakultät für Veterinärmedizin der Ankara-Universität reichen mehr als 60 Jahre zurück. Die ersten Kontakte gehen auf die 1960er Jahre zurück. Vor allem Professor Lies von der Institut für Virologie der TU und sein türkischer Kollege Professor Dr. Selahattin Gürtürk pflegten einen engen Kontakt. Bereits zu Beginn der 1970er Jahre gab es von der türkischen Seite unter dem Dekan Professor Dr. Satı Baran eine offizielle Anfrage für eine Partnerschaft an die TU. Nach dem ersten Informationsbesuch in Ankara wurde eine Dreierkommission gebildet, neben der Professor Lies als Partnerschaftsbeauftragten Professor Hans-Peter Salman aus dem Institut für Physiologische Chemie und Professor Dr. Wolfgang Bisping aus dem Institut für Mikrobiologie angehörten. Eine besonders enge Beziehung zur Türkei gab es schon früh, auch durch Professor Dr. Rudolf Schwarz aus dem Anatomischen Institut. Es sollte noch einige Jahre dauern, bis ein Regierungsabkommen zwischen der Bundesrepublik Deutschland und der Republik Türkei unterzeichnet wurde. Im Rahmen dieses Programms wurde im Anschluss an die technische und materielle Unterstützung für die tiermedizinische Ausbildung und Praxis das Abkommen für das Kooperationsprojekt der Ankara-Universität mit der Veterinärhochschule Hannover am 7. Januar 1981 im Amtsblatt veröffentlicht und trat damit in Kraft. Die Laufzeit der Vereinbarung, welche 6 Jahre gültig war, wurde regelmäßig verlängert. Mit diesem Projekt sind Mitarbeiter, Studenten und Kommissionen gegenseitig nach Ankara und Hannover gegangen und haben wichtige Arbeiten geleistet. Im September 1981 wurde auf einer Pressekonferenz in der Veterinärmedizinischen Fakultät Ankara-Universität der Zweck der Kooperationsprojekte und sein Beginn zum 100. Geburtstag von Atatürk unterzeichnet. Zunächst gingen im Rahmen dieses Partnerschaftsvertrages 3 türkische Tierärzte-Assistenten, Doktoranden für die Doktorarbeit nach Hannover. Inzwischen können von beiden Hochschulen in den kommenden Jahren zahlreiche Studierende als Austauschpraktikanten, Praktikantinnen und mehrere junge Wissenschaftler, Wissenschaftlerinnen an der Partnerhochschule arbeiten. Außerdem wurden zwischen den Jahren 1981 bis 2008 insgesamt 17 türkischen Forschungsassistentinnen in der Tierärztlichen Hochschule Hannover promoviert. Mehrere gemeinsame Forschungsprojekte und Publikationen waren die Ergebnisse dieser Besuche. 19 von 22 vorgeschlagenen Projekten wurden im Rahmen des Kooperationsabkommens akzeptiert. Aus den Themenbereichen der 19 Projekte erschienen türkische und fremdsprachige Publikationen von hoher wissenschaftlicher Qualität. Im Rahmen der Projektarbeit wurde auch in der Klinik für innere Krankheiten der Haustiere ein modern ausgerüstetes klinisches Labor für das Blut-, Harn- und Panzersalzuntersuchungen eingerichtet. Durch Ausstattung mit der moderner Laborausrüstung in der Veterinärmedizinischen Fakültät der Ankara-Universität wurde das Forschungslabor zur Verfügung gestellt. Mit Hannover verbinde ich persönlich weit mehr als eine Stadt oder Hochschule. Ich war Mitglied der ersten Doktorandengruppe aus Ankara, die an dem Austausch teilnahm. Meine Doktorarbeit fand an der Klinik für Rinderkrankheiten statt, aber wir konnten auch an spannenden Vorlesungen und Untersuchungen kranker Tiere teilnehmen. In der Klinik für Rinderkrankheiten begann die klinische Untersuchung jeden Tag um 7 Uhr. Professor Dr. Matthias Stöber sagte damals immer zu uns, Morgenstunde Gold im Mund. Um 11 Uhr begann in der Klinik die Quote, in der die erkrankten Rinder und Kälber von der Studierenden untersucht und behandelt wurden. Ein praxisnaher Unterricht wie dieser erfreute auch die Studierenden. Wir sahen in Hannover eine sehr moderne, gut ausgerüstete Hochschule, aber die Promotion gab uns alle Selbstvertrauen, interdisziplinäre Forschung, Teamfähigkeit und Kooperationsbereitschaft. Wir konnten großartige Professorinnen, Kolleginnen und Studierende kennenlernen. Mit sehr vielen von ihnen waren wir jahrelang befreundet. Diese Ergebnisse unserer Doktorarbeit waren unvergesslich. Außerdem war ich inzwischen noch 3-mal in den verschiedenen Zeiten in Hannover, um über die Kälberkrankheiten Vorträge zu halten. Sie sehen im Bild Theo im Jahre 1983 mit Dr. Asmus nach der Promotion und auch im Ausländeramt in Theo im Jahre 1983. Hanna Richter war damals da. Ausländeramt in Theo wieder 1983 zusammen mit von der anderen Ländern kommenden Doktoranden und meine Frau und Sabriye Doğanay und eine Studentin. Wir haben natürlich über 40 Jahre über die Kälberkrankheiten hier auch in der Türkei gearbeitet. Über diese Probleme haben wir etwas neue Entwicklungen, über neue Präparate verwendet. Und nach der Behandlung sehen Sie diese Kälber auch wieder aufstehen. Professor Dr. Bernd Lepenies 1988 und Professor Dr. Hans-Peter Salman 1992 haben die internationale Anerkennungen und ihre vielfältigen Arbeiten durch die Verleihung der Ehrendoktorwürde durch die Universität Ankara zum Ausdruck. Beide waren sehr vielbewusster, zurückhaltender, dabei aber offener und geradliniger, liebenswerter und intelligenter Mensch und wertvoller Ratgeber. Als erste offizielle Amtshandlung zeichnete Präsident Dr. Greif im Jahre 2002 Professor Dr. İbrahim Burgu von der Tierärztlichen Fakültät der Universität Ankara mit der Ehrenbürgerwürde der Tierärztlichen Hochschule aus. Burgu bekam diese Auszeichnung für seine Verdienste um die Hochschulpartnerschaft zwischen Hannover und Ankara, die besonders durch einen lebendigen Austausch von Wissenschaftlerinnen und Wissenschaftlern sowie Studierenden auszeichnet. Die Partnerschaft mit Ankara ist ein Beispiel für eine über lange Zeit funktionierende und fruchtbare Kooperation. Es besteht der gegenseitige Wunsch, dass sich dies auch in Zukunft fortsetzt. Bei der Entwicklung der wissenschaftlichen und technischen Zusammenarbeit hinsichtlich der deutsch-türkischen Beziehungen spielen die Tierärzte eine wichtige Rolle. Wir müssen dafür sorgen, dass wir unsere Beziehungen weiterentwickelt werden. Möge diese gewachsene Freundschaft und auch weitere junge Studentinnen und Studenten in unseren Ländern erfassen und über viele Jahre hinaus lebendige Erinnerungen und Beziehungen ermöglichen. Abschließend sei allen Kliniken und Instituten der TIO, die sich in all den Jahren an der Aus- und Weiterbildung der türkischen Studierenden und Wissenschaftlerinnen beteiligt haben und hoffentlich noch lange tun werden, ganz herzlich gedankt. Vielen Dank für Ihre Aufmerksamkeit.
Okay. Professor Şahal, thank you for your presentation. And we actually, at the current moment, we are expecting Professor Manfred Kietzmann, but I guess that Manfred Kietzmann will be coming to the closure session with Dr. Lepenies at 2:20 German time and 4:20 Ankara time. So at this moment, we can start. We're a little bit early, but we can start with the presentations of the session 1, the infectious disease session. And the first session is with Professor Dr. Aykut Özkul about the diversity of zoonotic arboviruses in Turkey. Professor Özkul.
Thank you very much, Begüm. I hope everyone is hearing me at the moment. There's no problem in hearing me. It's okay. So good morning, everybody. And first of all, I want to express my satisfaction to be here again with colleagues, even around the virtual roundtables. Let me open my presentation. Is that okay? Everybody can see the presentation?
Yes, we can. Perfect.
Okay, okay. So, dear President of TIHO and dear Dean and dear colleagues, it's very great satisfaction again to be with you all, with friends from Hannover after such a long time. So the main purpose of my presentation is to make a little open the door in a little area to re-stimulate the mutual collaboration between the biology institutions of both institutes. So there are a lot of biological related research already being carried out in the Ankara University in the Department of Biology, but today I would like to put a new focus on the arboviruses in Turkey, but these arboviruses having a public health concerns. As everybody well known, the arbovirus classification consists mainly 3 types of RNA viruses. These are positive, negative sense RNA viruses and double-stranded RNA viruses. But today, I would like to focus on flaviviruses and buniovirus, which are already being vectored by different arthropods in Turkey and in the field. And mainly all the data that I will share here is produced by our little research team in the field of Turkey. The first group of the viruses has been detected in the mosquitoes. The other one is in sandflies, and the last one in, of course, ticks. So I would like to kick off with the mosquito-derived flaviviruses. As everybody appreciates, I'm sure the West Nile is one of the biggest problem around the area in which Turkey has already been. And we carried out a lot of scientific papers on this infection. But today's presentation is mainly focused on the viruses that we detected in the vector arthropods. I would just mention these viruses that we detect in different mosquitoes. The most recent data that we observed in the field about the West Nile virus is detection of this virus genomic RNA in different mosquito pools that have already been collected in different parts of the Turkey. Mainly, we detected this virus in Operosauros caspius and Culex pipiens subtypes of the mosquitoes, and we defined this mosquitoes by morphological evaluation and also DNA barcoding. As you see here in the presentation, the different types of these mosquitoes as infected with this virus, about 2% in the field, depending on the time that we tracked these mosquitoes, because the collection of the mosquitoes is performed in different time points in the year, and mostly of the collection was done in especially late summer. So depending on the collection time, the percentage of the genomic RNA presence can be different. So as you see, the West Nile genomic RNAs detected this mostly in lineage 1 group of West Nile viruses. But I'm sure the colleagues interested with this virus has already aware that the lineage 2 virus has already been detected, but not in Turkey, but not in mosquito species. Beyond this virus, there are other flaviviruses has already been detected in mosquitoes, and most of these viruses has been related directly to the this arthropods. You see, there are some Culex and Theileria flaviviruses has already been detected, and some Operosauros caspius flaviviruses detected, and Anopheles originated flaviviruses has already been detected. We also detected with the novel flaviviruses has not been identified yet using the NGS systems. I want to skip another vector species, which are sandflies in Turkey. There are a lot of research has already been performed in sandflies. Our group and also some other research groups in Turkey, you will see there are some local flaviviruses has already been isolated or detected in the different sandflies in Turkey, and most of them have already been identified with their local original name and accepted in the International Virus Taxonomy Committee, and their localization mainly in the Mediterranean, Middle or Aegean Anatolia, and some of those has been found that already related with the disease in human being, and some of has still remaining a big question marks about the human public health concern. The Toscana viruses has first detected in Turkey in 2010, and there are a lot of reports on this virus infection in human beings so far, but especially sandfly fever Turkey virus is more important for us, because It causes central nervous system infections characterized by encephalitis and also more clinical symptoms when compared to classic flight fever that we see in southern part of European continental, especially in Italy, France, and Spain and their islands in the Mediterranean. And these diseases are characterized by the leukopenia, thrombocytopenia, and elevation in the different liver enzymes. The flaviviruses that we detect in Turkey are showed in their different genetical subsets in the molecular epidemiological analysis, especially the proscanoviruses, original proscanoviruses that we detect in Turkey found very closely related to the Zerdali virus. Zerdali is the name of the fruit in Turkey, and this virus isolated in the East Anatolian part, and Adana and Edirne viruses is also found very closely related to each other, although they are localized in a different geographic part of the Turkey, one in the Thrace, the other one in the Mediterranean Turkey, and also Taurus virus is localized in the different subsets. As a result of field study of my two PhD students, they found these flaviviruses can be found in the different animal species, especially Proscona virus and Cecidian virus in dogs was one of the most interesting one for us. The virus of that he detected in the Mersin and Adana region, and divided the two different genotypes or biotypes in the same phylogenetic analysis. And the other very interesting point of this observation is the viruses are together with the leishmania in the same dog. In another study, the other PhD student has detected the same viruses in the brain of the migrated birds. As you may know, Turkey has been settled in the two different major migratory bird routes, global bird routes, and just because of this reality, it's very likely to found different viruses and their different genotypes in the field. So the animals that he collected are those that handled in the rehabilitation centers in the north and the south of Turkey, and based on the biologic analysis, he found two different types of the same flavivirus in brain of the stork, flamingo, and pelican, and it's so clear that the one of those is quite different than the other two. The other arthropod that we focused for looking for different viruses in the field is ticks, because you know, Turkey is one of the leading countries in the Crimean Congo hemorrhagic fever, and we are reality to facing in increasing numbers sometimes. For example, this year and year before, we have records in the case of Crimean Congo hemorrhagic fever in human beings. So we collect a lot of tick samples from field, and beside of the Crimean Congo hemorrhagic fever viruses, we had a chance to find different types of virus in the same samples. So one of those is the TAMDI viruses. TAMDI virus is a quite different virus, and it has been related with the some human cases, and these viruses in the ticks is really more than that we imagined before, or that we foreseen before, and they are really in abundance, so that they can genetically showing a large divergence in the phylogenetic analysis. So based on these studies, the Anatolian tick flavivirus type 1 and type 2, and also Tachang tick virus 2 has already been detected in the local ticks that we collected in Turkey. Another virus that we found in Turkey is rhabdoviruses. I skip very fast these examples, but as you see in the slides, these are really very related with the well-known members of these viruses. One of the Rhipicephalus annulatus tick, we isolated Wuhan tick virus type 1, and this one also named with the WTV1 Anatolia, again in collaboration with the Federal Institute in Berlin. And tick2 viruses also detected in the same pools by the NGS analysis. One is Hyolama aegyptum, and the other one is the Haemaphysalis pulva. We detected the different viruses in these ticks, but these are really very important again for us as all tick virus 3 Anatolia and Chapping tick virus 3 Anatolia as two example, but important example of this virus. Q viruses is really very important because they are really very strange viruses, because most of the viruses can be classified segmented and unsegmented RNA genome. Two virus members exhibits in different types of RNA structure in their structure. Sometimes you can find circular, sometimes you can find segmented viruses in the field. So they are really very strange viruses. And the final part of my talk, I want to give brief information about the Jingmen virus, again in tick of tick origin. Jingmen virus is first identified in China in 2014, and then they have reported in different types of the human disease in 2019. Some of these diseases are severe febrile illness, and then people showed some skin findings. In Turkey, in our laboratory, the Jingmen tick virus luckily isolated from a tick homogenate in the Vero cell line. You will see here the CP of this virus on day 8 in the second passage on Vero cell line. And after sequencing the initial NGS applications, we designed different primers to look more detailed their epidemiological features based on its genomic structures. As you see, the local Jingmen viruses has already been detected approximately 5% of the tick pools in Turkey, and they are placed in two different genomic subsets in the molecular epidemiological analysis. So one of the most important maybe observation is these viruses is more related to the viruses detected before in Balkans, especially in Kosovo. So maybe this information is indicates that these kind of viruses is distributed geographically by the help of the migrating birds in Northern Europe over to Turkey and to Africa. And so all these abundance of the different types of the viruses led us to conclude that many viruses in different structures and characteristics have been described in arthropods. So this also indicates important potential to facing any time with the emerging new types or subtypes of the virus having veterinary or public health concerns in the region. So most of these or some of these pathogens or viruses are confirmed or probable pathogens with different viruses, diseases, and we are still focused on these viruses, and we are trying to enlarge our information capacity on these viruses because the information of their biological characteristics and epidemiologic dynamics is still very limited. So we are still working on these viruses in our research team, with our research team. So at the end of my talk, I would like to mention once again my satisfaction having touch again with our colleagues. So maybe this presentation shows our willingness to restart the collaboration of at least institutions of the both sides of the virology department or some different people. I don't know how it works or how it longs, but we have to do this. We have potential, you have potential, so we have to match them to make a big sparkle. Thank you very much for your attention.
Right. Thank you very much, Professor Aykut Özkul, for this great presentation. Okay, Dr. Nüket, please, if you have a question.
Thank you. I have a question. Thank you, Professor Özkul. According to phylogenetic analysis, we see that the viruses from isolated in Turkey located in separate branches. And what do you think that the common mutation drivers for the viruses? Could you make some comment on that? Thank you.
Yeah, this is another point of our concerns. It's very important because these viruses have a big biologic life in the nature and different arthropods besides vertebrates. So some of our colleagues is well experienced on these vector systems biology, so we are collaborating them. And most of these mutations, I'm sure they're originating their biologic life in these vectors, and it is very likely to make mixture of the viruses at being at the same time in the same vector. So it takes time. Most probably you will see in the slides, because we found some footprints of the recombinations in the genomes that we analyzed. So most probably these happens in the vectors and different types of the molecular interactions.
Okay. Once again, thank you very much, Professor Aykut Özkul, for your
You're
presentation.
welcome. It was a pleasure.
And now I'd like to present Dr. Paul Becker about the virus
Hala
research.
Bey, internet gelsin mi bir de bakın. 88. Tamam.
All right. Dr. Paul, can you hear?
Yes, I can hear you.
Okay, perfect. So welcome back again. And Professor Becker.
Tamam Hala Bey. Asistan bekliyorum. Gelsene. Okay. No, that is something. I'll go with.
Okay, so you can start screen sharing.
Okay.
Yep, we can see you. The screen.
You can see me. Can you
Yes,
see the
exactly.
presentation?
We can see it.
Okay. So, merhaba. Good morning, dear colleagues. So, I visited Turkey several times. First time when I was a young student, it's about 35 years ago. And the last time, it's about 8 years ago. And there I visited the veterinary faculty in Ankara. And this was a nice and also very interesting meeting. And today we meet virtually. And I thank all the people involved in organization of the symposium. So there is a later pointer. Okay. So at the Institute of Virology in Hanover, so we have different kinds of research interests, and also we have many projects, and I will summarize this now in my talk. And one major focus is RNA virus evolution and genetic diversity of mainly RNA viruses, and this also includes the identification and characterization of novel viruses like atypical porcine pescivirus or hepativiruses and pegiviruses in cattle and pigs, to give a few examples. And this topic about RNA virus evolution is also related to virus discovery and to study the genetic diversity and host range of viruses. And so far, this is related to pestiviruses. This is also associated to our interests, so our research focus with regard to epidemiology, diagnosis and control of classical swine fever, which we are doing in the framework of the EU, so European Union and OIE Reference Laboratory for Classical Swine Fever. And pestiviruses, so they form one genus in the family Flaviviridae. So Flaviviridae was also one focus in the last talk of my colleague. And for quite a long time, there were 4 pestivirus species recognized, so bovine viral diarrhea virus 1 and 2, classical swine fever virus, and border disease virus of sheep and goat. And these are small enveloped viruses. And yeah, as I said, until 2017, there were 4 approved species, but due to an expanding genetic diversity and host range of pestiviruses, we proposed a new nomenclature in 2017, and we proposed a total of 11 established pestivirus species, and they are termed now pestivirus A, B, C, so until pestivirus K. And I want to highlight one of these newly established pesti species. This is pestivirus I, and these are these idine or idine-like viruses. And the idine virus was first described by my colleague T.Çiğdem Oğuzoğlu, and T.Çiğdem will give a presentation later this morning. And T.Çiğdem did a PhD thesis at the Institute of Virology in Hanover about 15 years ago, and this was also then a starting point of a collaboration, during which we jointly further characterized such viruses, so the pestiviruses, they experienced a further expansion of the genetic variability and also host range, and this resulted in another very recent proposal. Just a couple of months ago, we proposed an update to the taxonomy of pestiviruses, and so we again proposed additional species, in total 8 novel additional species, and these comprise viruses from sheep or from sheep and goat, and also this Lindar virus is from pig. And then there are other viruses with non-ungulate hosts, and these comprise viruses from various bat species, from rodents, from pangolin, and also from a whale. So in total, so we have now 19 pestivirus species. So with regard to these pestivirus, so these idin-like viruses, so they are quite widely distributed in small ruminants and cattle in Turkey, and in small ruminants they can cause some border disease-like symptoms, so characterized by fetal malformation or abortion. And we then studied in detail the antigenic similarity among pestiviruses, including this idin virus, and so the main result there is that this idin virus is antigenically most closely related to classical swine fever virus, and therefore this is a problem. So this could interfere with classical swine fever diagnosis, and this due to cross-reactivity in ELISA and also in virus neutralization test. And we performed also an animal experiment with this virus to collect reference material, and this was also a prerequisite to do such studies based on cross-neutralization test. And the main result of the infection experiment was that there are no clinical signs in pig. However, if such a virus would be introduced in the pig population, this may cause severe problems to cause, to exclude, to differentiate classical swine fever virus induced immunological reactions from serological reaction caused by these viruses. And such animal experiments are performed now in this new building. This is the new home of the EU reference lab for classical swine fever, and I show you also the person responsible for the classical swine fever lab in this building. So another complex are these projects on virus host interactions, and so these projects on virus host interactions include a focus on innate immunity and also on viral entry mechanisms. I will not go too much into detail in that. I only want to mention that in case we have cellular receptor candidates, we can then confirm these cellular receptor candidates also by CRISPR-Cas9 mediated gene knockdown. And I want to briefly mention that we also have established differentiated airway and intestinal epithelial cell cultures, and so So here I want to show you briefly the precision cut bovine lung slices. And here on the left you can see a bronchus. Red staining is beta tubuline. Here in higher magnification, these are non-infected lung slices, and here on the right you can see that we can infect these lung slices with bovine viral diarrhea virus, and then we can see the distribution of the infected cells. Another option is these air-liquid interface cultures, and here primary cells are first initially propagated, and then they are seeded on filters, and on these filters then they experience a further differentiation, and with such alicultures we can study infection from the basolateral side and also from the apical side. And we use this also to further study BVDV infection in the respiratory tract. And here on the left you can see apically infected cells, and here on the right so the infected cell cultures after basolateral infection. And one conclusion from this is that the basolateral infection is more efficient than apical infection, so this was quite unexpected. And we also then determined the viral loads in the supernatant of these infected cell cultures, and here we could see that BVDV is released exclusively from the apical side. So this means that the bovine airway epithelial cells are not a door for the virus to infect the host and immediately mediate a systemic infection, because the virus is not released via the basolateral side. Then we also have established more recently several types of organoids, and I show you here the example of porcine intestinal organoids. And if these organoids are embedded in matrixel, so then we have these basal out organoids. So the basal side is outside and the apical side is in the center. This of course would be difficult to infect such basal out organoids with viruses mainly infecting from the apical side. But if we suspend these organoids, then we can reverse the structure, and then we can obtain such apical out organoids. And you can see here that these apical out organoids can be infected by porcine rotavirus A and also by other enteric viruses, which we can and we can use these organoids now for further study of viral infections. So another focus at the Institute is to study virus infections in terrestrial and aquatic wild animals. And so, for example, we have identified Hepatzi, also novel pestiviruses, Punya viruses in wild ruminants, to give one example. And then we also have a major interest in studying virus infections in marine mammals like seals and porpoises. And today I only want to briefly summarize very recently obtained results of the detection of influenza viruses in seals. And so in August this year, so just a bit more than about 3 months ago, so we detected highly pathogenic avian influenza viruses of the subtype H5N8 in 3 dead harbor seals at the North Sea coast in Germany, and this was the starting point of a collaboration with the Institute of Pathology, with the Institute of Terrestrial and Aquatic Wildlife Animals, and also with RITS, and at TIHO, and also with the Friedrich Loeffler Institute at the island of Reims, and so complete genome sequences were obtained and then compared with other influenza viruses, and we can conclude from this data that the 3 seal viruses are closely related to avian halopathogenic influenza viruses, and so the highest genome loads. This was interesting and also unexpected. So these were found in the brain, and so this high amount of influenza virus genomes in the brain. So this also correlated with pathological lesions and also with immunostaining of the influenza virus nucleoprotein in cerebrum and also in cerebellum. And in contrast, in lung and swab samples, we could detect only low or even no influenza A virus genomes. And in 2 virus isolates, we also have identified an adaptive mutation in the PB2 segment, and at a position which was implicated, which was reported before to be implicated in avian to mammalian host switch. Then in a follow-up study, we also investigated serum samples from healthy seals from Germany and also from the Netherlands, and so these samples were derived from the time period shortly after we've detected these HPAIV in the 3 seals, and so we used virus neutralization tests and also an H5 specific antibody ELISA, but using these methods, so we can conclude that there is no evidence for the presence of H5 influenza A viruses in these healthy seals. So the last point I want to mention here, this is that we have also a focus on vaccine development, and in the past we have focused on the generation, on the establishment, development of classical swine fever marker vaccine candidates, and today I do not want to comment on this, but I want to mention that we will have later a talk by Asisa Volz on the development of a COVID-19 vaccine based on the modified vaccinia virus strain Ankara platform. And so these different research projects, so on virus evolution, also on swine fever, on this virus-host interaction, to study virus infections in wild animals, and also these projects on vaccine developments. So they are all under the umbrella of the One Health approach. And with that, I want to close my talk and thank all my collaborators at the Institute and also at other institutions at the TIHO, and also collaborators from other institutions, and of course thanks also to the organizations providing funding, and thank you for your attention.
Okay. Professor Paul Becker, thank you for your presentation. Actually, we're quite out of time, 7 minutes past, but we can accept one quick question, if there is any from the audience. I don't see any in the chat session. Well, okay. In this case, again, thank you for this great presentation, Professor Paul. And moving to Professor T.Çiğdem Oğuzoğlu with her presentation on the expression of SARS-CoV-2 envelope protein in baculovirus system as a vaccine candidate. Professor Çiğdem, yeah.
Thank you very much. One moment, please. Can you see? Yes, I think. Okay. Okay. Meine sehr geehrten Damen und Herren, liebe Kolleginnen und Kollegen, ich grüße Sie ganz herzlich aus Ankara. Zuerst möchte ich mich herzlich bedanken, wer uns diese Gelegenheit gegeben hat. Es ist sehr schön für mich, mit allen nochmal hier wieder treffen können. Pandemie war ein Thema für uns, das wir zu unseren Studenten an der Virologie-Vorlesungen erzählten und die Beispiele aus der historischen Virologiegeschichte gegeben hatten. Leider, wir haben es als ganze Welt erlebt, die wir niemals gewünscht hätten. Heute wollte ich als eine Projektleiterin Ihnen von dieser aktuellen Thema etwas erzählen. Von TÜBİTAK finanziertes Projekt heißt Expression von SARS-CoV-2-Hüllprotein in Baculovirus-System als einen Impfkandidat. Wenn Sie mir erlauben, bevor ich meinen Vortrag halte, möchte ich auf Englisch eine kurze Erklärung machen. Ladies and gentlemen, dear colleagues, it is for me a great pleasure to be able to talk to you from here. For us, pandemic was just a topic which we told to our students on virology lectures, and we given examples from history of virology. We never wanted, unfortunately, we have experienced it worldwide. My presentation is today about SARS-CoV-2 infection and our project, and I will present in German language with English spelling. Neben der Nutzung vorhandener Impfstoffe sind Weiterentwicklung von wirksamer und sicherer neuer Covid-19-Impfstoffe zählt man zu den sehr wichtigen Faktoren, um anhaltenden Pandemie zu beenden oder SARS-CoV-2-Infektionen langfristig zu kontrollieren können. Impfen ermöglicht es, sowohl einer Verhinderung von Ausbreitung des Virus begegnet werden als auch mögliche gesundheitliche Ergebnisse einer Erkrankung vermindert und eine Belastung des Gesundheitssystems verhindert werden. SARS-CoV-2, wie wir alle kennengelernt haben, wurde als ein neues Beta-Coronavirus im Dezember 2019 in der Stadt Wuhan, China, identifiziert und Infektion wurde mit der daraus resultierenden Krankheit, die als Coronavirus-Erkrankung 2019 Covid-19 ist, bekannt. Coronaviren sind RNA-Viren und gehören zur Ordnung Nidovirales. Die enthalten 4 strukturelle Proteine, Spike-Protein, Envelope, auf Deutsch Hüllprotein, Membranprotein und Nukleokapsidprotein. Es gibt neutralisierende Antikörperbildung gegen alle strukturelle Proteine des Virus. Die Bedeutung der Impfung im Kampf gegen die Krankheit ist sehr groß. Der Rückgang der Infektionsrate nach der Impfung zeigt uns, dass es ein effektives und richtiges Weg für Krieg gegen Infektion ist. Es gibt in der Türkei, wie hier gesehen haben, ein Vaccinationsprogramm für Covid-19-Infektion. Bisher wurde über 75% von Totalbevölkerung für Gemeinschaftsimpfung, Immunität und geimpft. Manche Familien der Nidovirales, zum Beispiel Coronaviren, unterscheiden sich hinsichtlich der Struktur und Funktion der viralen Replikase von allen anderen RNA-Viren. Diese Funktion erhöht die Mutationsfähigkeit des Virus. Durch Mutationen sind verschiedene Varianten des SARS-Coronavirus-2 entstanden. Varianten wurden unterteilt, welche Aminosäureaustausche im S-Protein aufgewiesen sind, besorgniserregende Varianten und stehen unter besonderer Beobachtung. Wie hier alle gesehen haben, wir wissen jetzt, dass alle besorgniserregende Varianten am ganzen Welt verbreitet sind. Wir haben auch ein Projektvorschlag zur Entwicklung von Impfstoff für diese wichtige pandemische Infektion gegeben. Und wir haben in diesem Projekt Hüllproteine ausgewählt. Was für ein Protein ist Hüllprotein von SARS-Coronavirus-2? Welche Eigenschaften hat? Ich möchte hier ganz kurz zusammenfassen. SARS-Coronavirus-2 Hüllprotein ist ein kleines strukturelles Protein, das in vielen Aspekten des viralen Lebenszyklus beteiligt ist. Zum Beispiel dieses Protein ist verantwortlich von Pathogenität und von Virulenz. Dieses Protein hat Viropolin-Effekt. Es ist ein kleines hydrophobes Protein, das Ionkanalaktivität zeigt. Und besonders wichtig für uns, diese Stabilität gegen Mutationen. Es ist die E-Proteine von SARS-Coronavirus-2 und SARS-Coronavirus-1 unterschieden sich nur in dieser Region durch 3 Substitutionen und eine Deletion. Diese starke Homologie unterscheidet dieses Protein von anderen und sorgt für Stabilität dieser Domain. Für SARS-Coronavirus-Stämme, einschließlich aller Varianten, wurden keine Änderungen festgestellt vom Wuhan-Stamm bis heutige Stämme. Was haben wir geplant und welche Schritte hat unser Projekt? Im ersten Schritt, wir haben Hüllprotein von SARS-Coronavirus-2 Ankara-1-Stamm gewonnen und im Transfer plasmiert kloniert. Danach, es wurde Expression von rekombinanten E-Protein im Bakulovirus-System in Karadeniz Technische Universität gemacht. Und als letzte Schritte, präklinische Studien haben wir hier gemacht. Kontrollierung der Immunantwort gegen rekombinantes E-Protein SARS-Coronavirus-2. Diese klinische Experimentelle haben wir an unserer Abteilung durchgeführt. Wir haben unsere Experimente beim IFNAR-Mäuse gemacht und zuerst haben wir diese Tiere geimpft, zweimal geimpft und danach Challenge-Virus gegeben. Unsere vorläufigen Ergebnisse sind gut und wir haben neutralisierende Antikörper gegen das erhaltene rekombinante Protein festgestellt. Unser Studium ist laufend. Die waren unsere vorläufigen Ergebnisse. Und es dauert noch, aber wir haben Hoffnung. Ich möchte hier mich ganz herzlich bedanken, meine Kollegen aus Ankara-Universität Veterinärfakultät und aus Karadeniz Technical Universität, die in unserem Projekt zusammengearbeitet haben. İlke Karayel Hacıoğlu, Ali Rıza Hanife Necat, Aykut Özkul, Zihni Demirbağ, Saber Delpasant Kabbasi und TÜBİTAK unterstützt uns als finanziell. Und besonders möchte ich unsere Doktoranden Ece Adıgüzel, Eda Erdem, Nazlıcan Filazi, Katalin Földes, Buket Gül, Zehra Fırat Karagöz, İlker Şahin Kesen und aus Karadeniz Teknik Üniversitesi Associated Professor Dr. Hacer Muratoğlu herzlich bedanken. Es gilt meine Danksagung. Erstens möchte ich meine Chef und Professor Dr. İbrahim Burgu ausdrücken. Hat in den bilateralen Beziehungen zwischen Ankara und Hannover sehr viele gegeben. Deswegen sein Name möchte ich hier erwähnen. Zu guter Letzt möchte ich im Respekt meinen Kollegen aus Hannover auch erinnern. Professor Möning war, hören Sie mich, eine andere, Asisa Volz. Ja, genau, Moment. Ich muss noch mal einen Moment. Ich möchte diese Bilder zeigen, deswegen, es tut mir leid. Ja, genau. Mein Doktorvater war Professor Möning. Ich möchte ihn auch ganz herzlich bedanken. Und ich möchte auch in Respekt Dr. Frey, Dr. Greiser-Wilke und Dr. Haas auch herzlich bedanken. Die sind jetzt nicht mehr da, leider. Und meine Kameraden, meine Mitdoktoranden waren dort damals immer zusammen. Ich möchte auch herzlich nochmal bedanken für Ihre Aufmerksamkeit. Dankeschön.
Okay. Professor T.Çiğdem Oğuzoğlu, thank you for your presentation. If you'd like to have a question, we have time for one quick question. Or we will be moving with our next speaker. Okay, thank you very much, Professor Çiğdem. Now we're moving with the next speaker, Professor Asisa Volz, on her presentation on immunogenicity and the efficacy of the COVID-19 candidate vector vaccine, MFA SARS-2S, in preclinical vaccination. Okay.
Can you see my presentation?
Yes, we can. Perfect.
Oh, this is nice. So thank you for the kind introduction. And it is my particular pleasure to participate in the Hannover Ankara meeting. And also because my research, as you will learn in a few minutes, is kind of directly connected to Ankara. So, and a major focus of my research is the development of improved vaccines against zoonotic infections. And this is also a central part in the One Health approach. And in the One Health approach, the so-called new emerging zoonotic pathogens are especially important because they can induce really severe and lethal disease in humans and can rapidly spread all over the world, as of course we see it now with the SARS-CoV-2 pandemic, but this is important. We have that really in a regular time schedule. So remember the Ebola epidemic in 2014. And this is exactly the area where we want to contribute with our vaccine development. And now you will learn about my connection to Ankara, because our vaccines are based on the modified vaccinia virus Ankara, a highly attenuated and replication deficient vaccinia virus, a vaccinia virus you might all know as the prototype of a live viral vaccine that has been successfully used to eradicate human smallpox. And MVA, the modified vaccinia virus Ankara, has been developed in Munich at the veterinary faculty already in the 1960s by Anton Meyer. And Anton Meyer received the ancestor virus of MVA from the State Vaccine Institute in Ankara, based on his long-standing collaboration with the Vaccine State Institute. And he used the vaccinia virus strain Ankara for serial passages in chicken embryo fibroblasts. And after more than 500 passages, the vaccinia virus Ankara appeared modified in its phenotype. And so Anton Meyer named it modified vaccinia virus Ankara, short MVA. And that modified phenotype included a really nice phenotype, meaning that MVA cannot productively grow in mammalian hosts. And what does that mean in detail? You can see that here. So MVA can enter the cells, can start its replication cycle with unimpaired gene expression, but then here, at that step, MVA fails to produce new infectious variants. And so MVA cannot spread within mammalian hosts and cannot cause disease here. And so MVA has been established as safe viral vector for insertion of foreign genes and for the generation of recombinant vector vaccines against various infectious diseases. So, and we used MVA in many different approaches, and quite promising was our approach to generate an MVA-based vaccine against another zoonotic beta coronavirus, against MERS, the Middle East Respiratory Syndrome coronavirus, because here we established.
Excuse me, Asisa, one can only see your first slide. You have to click on the additional slides. Or is that still your first slide? Slides do not move.
Now?
No, not yet.
No, started on the first slide.
Yeah.
Could you please presentation mode, move to presentation mode, maybe?
I'm in the presentation mode.
No, but in
if I can try to share the slides. You just click on.
I cannot see anything.
Okay, maybe I share the slide and
now
you
it's
just
moving.
do the presentation.
Can you move again?
That's okay.
Now it's fine. Yeah.
Şimdi gördüm.
I cannot see anything now. Now?
Yes, very good.
Okay. Now it's stuck again. Is it possible to do close your PowerPoint presentation, open again, and close the stop sharing? Maybe it's much better.
Is that now from?
Now it's closed, and then you can start back again, opening the PowerPoint, opening your slide again, please.
Bernd, can you share the slides? I think this is much easier.
Yeah, I can share the slides, and you just maybe that's the easier way. So just. Okay, you should now see my screen.
Yeah.
Yes.
Now it's fine.
Which slide should I click on? And then you just say next slide, so that should be fine.
I think go to the MERS slide.
Yes. Good.
Yes, this slide. So then back again. Sorry for the delay. So we used here the MVA for the establishment of a technology platform to rapidly generate vaccines directly from animal models to clinical testing. And the MVA MERS-S vaccine developed in that project has been also successfully tested in the first phase 1A clinical study. And here MVA confirmed to be very safe in the humans and also highly immunogenic. So, and we also found that a third application of that MVA MERS-S vaccine 1 year later even significantly increased the levels of neutralizing antibodies, and this looked quite nice and quite promising. And with these very important data in the beginning of 2020, we were already faced with the emergence of the new coronavirus, the SARS-CoV-2. And so we decided, based on that very promising results from MVA MERS-S, that we, within the DEZIF, the German Center for Infection Research, we will use the MVA MERS-S strategy as blueprint for the generation of a COVID-19 candidate vaccine. And so we choose the unmodified spike protein as vaccine antigen, and we inserted the S gene encoding sequences within the MVA genome using our well-established standard methodology, also compatible with clinical evaluation. Next slide, please. And in vitro characterization confirmed the stable synthesis of a mature and fully glycosylated spike protein, as you can see in the upper right, in immunostaining of infected cells, and we also confirmed it in Western blot analysis down left. Yes, and then we also confirmed that MVA SARS-2S replicated to high titers in the main production cell for MVA vaccines in DF1 cells, and as you see below, MVA SARS-2S still maintained its replication deficiency in human cells. So next slide, please. Then we went on to the more exciting part, to the in vivo testing, and here we vaccinated bulb C mice intramuscularly based on our MERS experience, and comparing two dosages, 10 to the 7, 10 to the 8, and as you can see, two dosages induced neutralizing antibodies and even to higher titers after the boost vaccination. And we were also really happy that we also confirmed the activation of S epitope specific CD8 T cells, as you see below, in ELISpot analysis and FACS analysis. Then for the next step to test the efficacy, next slide, please. This is not so easy in the mouse model because mice are normally not susceptible for SARS-CoV-2. And so we used a special SARS-2 mouse model where mice got susceptible for SARS by transduction of the specific ACE2 receptor using adenovirus vectors. So we again vaccinated bulb C mice, prime boost schedule, and 2 weeks after the last vaccination, all mice were intranasally transduced with an adenovirus vector expressing the ACE2 molecule. And 3 days later, we infected all mice with SARS-CoV-2 BAFPAD1 isolate. And as you can see below, the control mice that got PBS for vaccination mounted high levels of virus in the lungs. And the nice result was that we did not detect any SARS-CoV-2 virus in the MVA-SARS-2S vaccinated mice. So next slide, please. This is then also confirmed in pathohistology, as you can see in the lower lane, where we have the control mice. Here we detected large areas of densely packed inflammation in the lung and also large areas of virus antigen. So next slide, please. So we went then on with our characterization and tested the Syrian hamster model. And the Syrian hamster is now also from the WHO considered as the best animal model to mimic COVID-19 from humans. So we again performed prime boost vaccinations, and 4 weeks after the last vaccination, all hamsters were intranasally challenged with SARS-CoV-2. And as you can see here, 3 to 4 days after the challenge, all control hamsters started to lose weight and also developed clinical disease. And the nice result was that we did not detect that in our MVA-SARS-2S vaccinated hamsters. And we also detected high amount of SARS-CoV-2 virus in the lungs of the control hamsters, whereas we could hardly see that for the vaccinated hamsters. And this is also confirmed in the pathohistology, where you see in the lower picture the lung of a control hamster, and there you see really in the central part consolidated lung tissue, and the vaccinated hamsters look quite nice in contrast. So these results were quite promising. Next slide, please, and provided important preclinical data to work on our final goal to bring that vaccine into clinical use as soon as possible. And this is what we worked on in parallel within the DECIF consortium together with our industrial partner IDT Biologica. And IDT worked over summer 2020 on the optimization of their production process to finally obtain a highly pure MVA-SARS-2S drug product. And this has then been delivered to the DECIF clinical site in Hamburg under the lead of Professor Marilyn Eddo. And so we started in October 2020, next slide please, a first phase 1A clinical testing in 30 healthy humans. Again, we performed prime boost vaccination, compared 10 to the 7 to 10 to the 8 PFU intramuscularly, and the primary goal of that clinical testing was to evaluate safety and tolerability, and the secondary objective was immunogenicity. So, and preliminary data confirmed that MVA-SARS-2 is safe, so we observed only minor side effects around the injection site, but the first results from immune monitoring indicated that a further improvement of the vaccine is needed. And so we immediately started with the improvement of the MVA-SARS-2S vaccine. That means we optimized our S vaccine antigen, and we also revised the dosage of our vaccine formulation. These studies are now completed. The results look quite promising. Next slide, please. And we are now back in our clinical schedule. That means we continued the clinical testing in humans. In September this year, we started the next phase of clinical testing, the phase 1B, with the optimized vaccine, and we are quite optimistic that we now get better results. And for that short break we got through that optimization process, we see that now as an excellent training for future challenges in the case that there are new virus variants that induce another improvement of the vaccine. Next slide, please. So with that, I'm at the end. So I presented you the generation and characterization of the DCIF MVA SARS-2S candidate vaccine, and MVA SARS-2S confirmed to be immunogenic and protective in different animal models, confirmed the safety and tolerability in humans, and now we are quite happy to see the first results of the ongoing clinical testing. So, and next slide, please. With that, I would like to thank the consortium that worked on that really big project, and I would like to thank you for your attention and for your patience with the technical problems, and I'm happy to take questions.
Okay, thank you very much. Professor Aykut has a question.
Dr. Asisa, thank you very much for your very nice presentation, and congratulations for the achievements to produce mRNA-based corona as vaccine for human use. May I have a few questions in parallel, please? The first one, not with the SARS-CoV-2, but we also use the same platform for another virus before. What the biggest problem that we have faced here is about the promoters of the transgene. Could you please give some clues about the promoter that you use to express this virus? Is that synthetic or MVA promoters?
This is the modified, well-established classical vaccinia virus promoter as published from Bernie Moss. This is the PMH5 promoter.
PMH5, okay, okay. How about the transferability of the immunity in animals model during your immunogenetic testings either in mouse or in hamster models? Did you transfer the immune T cells to another animals to see this transferability?
So no, we did not do that because the mouse model is kind of the second choice of testing the vaccines, so COVID-19 vaccines, and especially in the context of that transduced model. So this is kind of only temporarily susceptible for the SARS-CoV-2 virus. So we have now here at TU established the K18 ACE2 mouse model, and that makes more sense to do some studies in that model because the other is just temporarily susceptible.
Okay, thank you. Maybe the last question. Did you check the duration of the immunity in the animals?
No, we did not test that.
And you terminated the assay in the 7th day or in which day?
Yes, absolutely.
Okay, thank you very much.
And even quicker for the mouse model.
Okay, thank you.
Okay. Professor Çiğdem, you can ask your question, or yeah. I guess there's a
Sesim geliyor.
Geliyor hocam.
Okay, okay. I would ask Asisa another question. How many microgram give you a mouse disease protein? Can I ask this?
This is not in microgram. This is PFU. This is a real live virus. So we use infectious units.
Okay, okay. Thank you very much.
Okay. Thank you very much, Professor Asisa, for your presentation. And we are now moving with Professor Valentin-Weigand with his presentation on how swine influenza virus and Bordetella bronchoseptica paved the way to Streptococcus suis infection in porcine respiratory tract. Hello, Peter, once again.
Yes, good morning, everybody, and thank you very much for inviting me to participate in this symposium. I have just shared my screen with you and hope you can see it.
Yes, we can.
Okay, yeah. In the next 10 minutes, I would like to give you an example of our research at the Institute of Microbiology. And you heard in the morning that Professor Wolfgang Bisping was one of the initial contact persons at the TO. So he was director of the Institute a long time ago. And now we are working on different pathogens in the Institute, and one is Streptococcus suis. And we are particularly interested in the last years in how swine influenza virus and Bordetella bronchoseptica, these two pathogens, pave the way. And what I will share with you today is an example of our recent results on that. And to start with the group, so this is the research group, and I would like to point out Maren Willenborg here. She's a former postdoc in my lab, and Desiree Bötsch, she's a PhD student and now also a postdoc in my lab. And these two were the main scientists who did the work which I'm showing right now, together with a cooperation partner in virology, but I will come to this at the end. Now, first of all, a very few words about Streptococcus suis, one of the pathogens we are studying. It's a pathogen in swine, it's occurring worldwide, and it's on one hand a commensal in the upper respiratory tract of pigs, but also it can cause invasive infections in pigs, causing meningitis, polyarthritis, septicemia, pneumonia. And it's also a zoonotic pathogen, which has caused some outbreaks, especially in Asia. There are several factors which are known causing virulence. One is the capsular polysaccharide, and you will see some slides on that later on. Now, we are interested in how Streptococcus suis is getting together with other pathogens in the respiratory tract, because this is the main niche of Strep suis. And within the porcine respiratory disease complex, as you know, the PRDC, there are, among others, two primary pathogens which can cause infections and also pave the way for Strep suis. And one is swine influenza virus, and the other is Bordetella bronchoseptica. And in the following, I will give you examples of results which indicate that both pathogens can, in fact, pave the way for Strep suis infections later on. The models we are using have been shortly already introduced by Paul Becher this morning. One are the precision-cut lung slices shown here, which is a very nice ex vivo system which can be used to study host pathogen interactions in a natural setting in the porcine respiratory tract. And the other model are the air-liquid interface cultures, which allow us to generate highly differentiated cells, especially the ciliated cells, which we can nicely see here on the HA staining and on the scanning electron micrograph. So these are the two models we are using. And in the first part, I would like to show some co-infections of swine influenza virus and Strep suis. And this is one example how we study this. This some imaging techniques, because we wanted to know how or whether SIV might facilitate adherence, so the first step of colonization of Strep suis. And what you see here is immunostaining of swine influenza virus H3N2, shown here in red. The second here shows you Strep suis stained in green, how they attach to bronchular epithelial and alveolar cells in the bottom here. And this shows you a non-encapsulated mutant of Strep suis, because it's generally assumed that the non-encapsulated bacteria attach better than the encapsulated, even though the encapsulated Strep suis is the wild type. So one speculation is that in the respiratory tract, Strep suis might downregulate the capsule expression and thereby better adhere. So what we did, we co-infected by first infection with H3N2, and then secondly by Strep suis, and then looked for attachment by imaging. And what you can see here nicely is when you look at the wild type strain, you have a clear enhancement of attachment shown in green, and it looks like Strep suis preferentially attaches to the virus-infected cells, which can be nicely seen here and down here, whereas this was not the case with the non-encapsulated Strep suis strain. And when we quantified this, That's one example. You can nicely see that the positive bacteria-positive cells increased significantly in the co-infection scenario with the encapsulated wild-type strain, whereas no such effect was seen when the bacteria were not encapsulated. So we speculated that the capsule of the bacteria is contributing to the facilitation of adherence by the swine influenza virus. And one model or one hypothesis, which is quite plausible and obvious, is that when you look at the respiratory cells and which are infected by the virus, by swine influenza virus, at a certain stage, these infected cells express hemagglutinin at the surface, and this hemagglutinin can be bound by sialic acid residues of the bacterial capsule. We have done also some other assays and tests to prove that there is a binding of the sialic acid residues of the bacterial capsule to these influenza virus hemagglutinin, either on the virion or on the epithelial cells. We have done some further studies which showed this. I will not show going into detail here, but I would rather show you a second step, which is also facilitated by swine influenza virus, and this is the effect of the SIV on cilia activity, which can be measured in such a model like the PCLS. What you can see here is when these epithelial cells are co-infected with influenza virus and strep suis again, within 24 hours up to 72 hours, you have a clear decrease in the cilia activity, and in every setting you have the same type of effect here, what you can see. When we did this with another influenza virus, H1N1, under these conditions, there was no such effect. So this allowed us to nicely show whether the cilia activity reduction induced by the influenza virus would have an effect on suis infection. And as a control, so to say, we had H1N1 because this was a virus which at these time periods did not reduce cilia activity. And when we then again did immunofluorescence imaging, we checked again by staining for influenza virus infection. And here what you can see here, the stain are the ciliated cell, beta-tubulin is stained here in red. You can see strep suis wild type and encapsulated mutant infection again. You see the wild type is attached to these surface epithelial cells, the ciliated cells quite poorly in the wild type, but much more in the encapsulated mutant, because the capsule is removed and allows expression of other adhesions. When we then co-infect with the virus strain H3N2, which has this drastic effect on the cilia activity, you can nicely see that in both strep suis strains, independent of the capsule expression, there seems to be a removal of the ciliated cell by the virus and an invasion of the strep suis cells a little bit deeper into the tissue. And this is not occurring when we co-infect with the H1N1 virus. Here, the strep suis stays on the surface, attaches to the surface, probably also to the hemagglutinin, but the facilitation of deeper invasion is only occurring in the H3N2, the virus which also reduces cilia activity. Now we can conclude that the SIV virus promotes adherence to an invasion of the PCLS by strep suis in a two-step process. One is initial capsule-dependent adherence to SIV-infected epithelial cells, so to the hemagglutinin. The other is a subsequent invasion of strep suis after SIV-induced ciliostasis. So this was what we found with the SIV. And in the second short part, I would like to show you our first results on the co-infection with Bordetella brachyseptica. So why did we choose Bordetella brachyseptica? The reason is that there are several reports, some of them are quite old, like you see 1989, for example, or here 1992, which showed that Bordetella brachyseptica is a pathogen which can pave the way for many other secondary pathogens, also strep suis, and in experimental infection of piglets with strep suis, it has been shown that if you pre-infect with Bordetella, you can get a better infection with strep suis. And it's also one hypothesis that Bordetella brachyseptica also reduces cilia activity similar to SIV. So we use it as kind of a model to check whether the action or the activity on the ciliostasis would also pave the way for strep suis infection in a similar way or similar to the findings on the SIV pre-infection. So first we did we had to establish the infection model with PCLS again. What you see here is an imaging of immunofluorescence staining of Bordetella ponticiptica in red beta-tubulin staining with ciliated cell. And here you see nicely in green, Bordetella attaches firmly to the ciliated cells, which can also be nicely seen here in this electron micrograph. The scanning electron microscopy shows you these Bordetella heavily colonizing the ciliated cells here. So we also checked the effects on ciliary activity and could show that dependent on the infectious dose here, 10 to the power of 2 up to 10 to the power of 4, that there is a clear effect on the ciliary activity, which drastically goes down after 24 hours already, and especially with the highest dose. So there's almost no ciliary activity after 48 hours. And in this model, we used the co-infection with Strep suis. So we first infected with Bordetella ponticiptica, similar to the SIV infection, and then did a secondary infection with Strep suis, then checked for the ciliary activity again. What you can see here is, here we started infecting with suis, and within these 4, 24, and 48 hours, ciliary activity was going drastically down. And in this scenario, we also checked the CFU counts, which are showing us the growth and the attachment, the association of Strep suis with the epithelial cell. And I would just like to point your attention on this effect here. Here you see the wild type Strep suis in the mono infection. Here you see the infection of epithelial cells pre-infected with Bordetella ponticiptica, and you see a clear increase here in the number of CFU which we plated from these studies. So we can say that Bordetella ponticiptica, probably by ciliostasis or reducing ciliary activity, is also paving the way for Strep suis. So these are studies which are just in the beginning, and we will continue this. We want to check and look for the mechanisms. But what we can say is, because this was interesting also, whether both pathogens attach to the same site. And it seems that they are not doing this, because what you see here, especially in this colorized electron micrograph, shown in red is Bordetella ponticiptica, in blue you see Strep suis. So they seem to attach to different sites. So they seem not to compete for colonization. And this explains why the reduction of ciliary activity is on one hand facilitating Bordetella ponticiptica attachment, but also co-facilitates the infection with Strep suis. Now, with this, I'm at the end of my presentation of our results. I would like to acknowledge funding. And funding of these studies has mainly been supported or done by the DFG and by EU Horizon project, the Global Prevention of Strep suis, abbreviated PIGS. I would like to acknowledge the people. As I mentioned in the beginning, Maren and Desi were mainly involved in these studies, but most importantly, this was a cooperation with the Institute of Virology of the TO, the group of Georg Herrler, and they initiated the primary or the initial co-infection study with the SIV virus. I would like to end, but at the end mention also that we have some other groups in our institute. So one group is working on mycobacterial infections. This is the group of Ralf Goethe. We have a group working on mycoplasma infections in swine mainly and in ruminants, which is led by Jochen Meens. We have a third group of Judith Bode, and she is mainly working on Brachyspira infection in swine. And last but not least, we have the group of Jutta Verspol, and she is mainly focused on diagnostic microbiology and especially on antibiotic resistance. And with this, I'm at the end of my presentation, and I would like to thank you for your attention, and I'm happy to take any questions if the time allows this. Thank you very much.
Thank you very much, Professor Peter, for your great presentation. Are there any questions from the audience? Okay. Well, in this case, I'm going to move with the next presentation. Thank you very much once again, Professor Weigand. And moving with our next speaker, Dr. Gökben Özbakış Beceriklisoy from the Department of Parasitology. She'll be presenting a retrospective review on zoonotic Toxocara in Turkey and Germany. Dr. Gökben.
Thank you, Dr. Begüm. Sorry. I think I share my slides. Okay. Thank you, dear colleagues and guests. It is my great pleasure to be a speaker of the 8th Ankara-Hannover Cooperation Symposium. Thank you for this opportunity. I have been to Hannover in the summer of the 2016. I had been a chance to work with the Professor Strube and her laboratories. It was great honor for me. I also applied as scholarship programs to being there again. The final decisions will be announced at the end of this month. I hope I get these scholarships to be a part of Professor Strube's lab again. Today I want to talk about the zoonotic Toxocara infections. Toxocara canis and also Toxocara cati are the worldwide distributed zoonotic roundworms, domestics and also wild carnivores, which may cause several forms of syndromes in the paratenic hosts, including the humans. It is a public health problem. Toxocara canis and Toxocara cati, one of the considered as the causes of the human toxocariasis. This systemic literature search was conducted in the Web of Science, Scopus, PubMed database, and also national Turkish indexes from 1000 to 2021 years records. The search strategy combined with the 4 items. First one is the prevalence of the Toxocara species in the definitive host, the domestics and also wild carnivores in the Turkey and the Germany. The second one is the prevalence of the natural Toxocara species infections in the non-human paratenic hosts. And the third one is the prevalence of the environmental contaminations of the Toxocara species, such as vegetables, sand beans, the soil contaminations, and also the cold contaminations of the domestics and also wild carnivores. Prevalence of the Toxocara canis is our fourth items. I'm sorry. The prevalence of the Toxocara species in the definitive host is examined with the necropsy for adult parasites and also by the stool examinations for the searching for the their eggs, and also sometimes they pass out the adult forms with their feces, fecal samples too. The prevalence of the Toxocara species in dogs and cats, especially there are various studies which are conducted in Turkey. The studies covered from each region of Turkey except the southern Anatolia region, and the prevalence range from 9.8 to 48.7 to dogs. And also for the cats, the fecal results of the fecal examinations, the prevalence is range from the 27.8 to 37.5 percentage in the whole studies. What about the wild carnivores in Turkey? There are only 3 studies were conducted in the wild carnivores, and all the whole animals are the red foxes, and also the Toxocara is recorded from the range 14.56 to 20% of the sample. What about the prevalence of the Toxocara species in the domestic animals of the Germany? There are various studies which are conducted in the Germany. In these studies, the considerable samples were collected to all of parts of the Germany. In the fecal samples, in the dogs, 2.82% to 22.4 for dogs prevalence, and also in the cats it occurs 3.9% to 26.2 with on the fecal examinations results, and with one postmortem examinations for the cats, and also their percentage is so high, it's 53% of results as we see in this table. What about wild carnivores? The prevalence of the Toxocara species in wild carnivores. The studies were conducted from the 5 various sources. In Turkey, as you know, there are just 3, and the whole samplings collect from the red foxes. For Germany, the various species can be seen, the raccoon dogs and also the grey wolves, the red foxes again, of course, and the stone martins too. And the eggs were ranged from the 5.19 to 13.4 in the fecal samples, and Toxocara also occurs 70.57 to 73 in necropsies in Germany. The prevalence of the non-human paratenic host, there are just two studies in the Germany. In these studies, they collect necropsy and they search for the skeletal muscles tissue and or brain tissue in the wild rodents. It has various sources, various species in the wild rodents, and these two studies were conducted in the Sorton of the Germany and also the Centrum of the Germany site. There are for the Berlin sampling, there are Toxocara canis larvae were detected in the 6 muscles and the 2 brain tissue, and also Toxocara cati larvae were detected in only 1 muscle tissue. In these studies, they can differentiate the Toxocara canis from the Toxocara cati with the molecular diagnosis to amplify of the ETS2 region. And the second study, they search various amounts of the samples, but it does not occur any Toxocara larvae in the Germany. And what about in Turkey? There is no record on the wild rodents in the Turkey. When Toxocara species eggs pass out of the feces, they release and contaminated to the environment with the roots. The humans and also the definitive host and also the paratenic host can infect with these roots. Because of that, some studies have been conducted on the cut of the definitive host, on the robe and also uncooked vegetables, and also the soil for the detections of the soil contaminations. For the contaminations of the cod in Turkey, there are only 4 studies applied in Turkey. The holes are, the samples are belonging to dogs and also the cats. And the Toxocara species embryonated eggs recovered from the whole of the dogs and all of the dogs and cats sampling. It is important for the contaminations route for the these infections, and also from these 4 studies, the hair samples were collected from the perineal region, caudal aspects of the hind leaves, and also the underside of the tail and back side of the body. What about Germany? Toxocara species contaminations of the dogs and cats. There is no new data from the Germany, but it's the first studies are published in the world in the Germany, 1973. In the dogs and cat frogs, and there is one embryonated Toxocara CPX on the cat's hair. Toxocara species contamination with the playgrounds and the parks. It's about soil contaminations. There is 12 studies were conducted in Turkey in that period, and when we check the results of the percentage, results of the Toxocara species eggs, it's similar results with the definitive hosts percentage in Turkey, as you see in here. The studies are conducted the 4th region of the Turkey. As you know, Turkey has the 7th region. There is no data from the 3 regions of the Turkey for these slides. And Toxocara eggs contaminations with the soil samplings. There is one study which is conducted in the Hannover University from Professor Strube's labs. In fact, Professor Strube's laboratory data. From these studies, 23.2% of Toxocara species eggs are recovered from the sand pits and also the playgrounds of the eggs. And the Toxocara species contamination with the vegetables in Turkey, there are just 4 regions conducted in this issue, and when we check the results of the studies, we can see the low percentage of the Toxocara eggs from the vegetables, the raw ones, and also when we check the Germany, there is no data available from the Germany's vegetables, the contaminated eggs. The conclusion, the differences could be related to broadly to sample size and also animal populations, laboratory methods, of course, the climatological variables, climate change in the regions, as well as the cities and regulations, and also the environments of the cities. We can prevent and also control this public health problems with the parasitic examinations should be performed regularly in the animals. In the infected animals, the deworming process should be applied, and also application of antiparasitic drugs should be applied in the stray animals. The dog owners and also sometimes cat owners collect the fecal samples from the parks, backyards, and also the playgrounds, and constructions of the special parking areas for pets, animals, and the public health educations. The humans should be informed on the potential for the infections. Thank you for your attention.
All right. Dr. Gökben, thank you for your great presentation. And we are almost out of our time, so I would like to move to our next speaker, Professor Dr. Christina Strube. She'll be presenting a neglected zoonotic parasitosis in neglected people. So, Professor Strube.
So, good morning to you all. Can you hear me?
Yes, we can hear you. Perfect.
Let me share my screen. Just one moment. Can you see my slides?
It takes like 5 seconds to see, all right? Now we see. Yeah, perfect.
Okay. Thank you very much. As Gökben already said, Toxocara worms or toxocarosis, which is the disease caused by these worms in humans, is a major parasitic problem. Nevertheless, toxocarosis is a neglected zoonotic parasitosis in the world, and especially in neglected people. And what I want to show you today is a study we conducted in such neglected people in an indigenous tribe, the Viva. We are doing a lot of work on toxocarosis. We are doing much molecular work in brain infection, but today I want to show you a epidemiological study in this neglected people. My slides do not move.
No, they're not moving. Did you make the full screen mode?
Yes.
Okay. Maybe you do the escape and do the full screen mode back again. Sometimes it is locked and it takes some minutes to take it.
Okay. So yeah. Full screen mode again?
Yes. And then...
now it's working. Perfect.
Yes, perfect.
Okay, as you already heard, we are dealing with two parasites, the dog roundworm, Toxocara canis, and the cat roundworm, Toxocara cati. And you also heard that humans can get infected, and depending on the organs which are affected, we can have visceral disease, we can also have ocular disease, which can lead to blindness, and we can have the so-called neurotoxicarosis associated, amongst others, with meningitis or cognitive deficits. There are three routes of infection for humans. The first route is the ingestion of embryonated eggs, meaning eggs containing an infective larvae. This is often the case in children with eating soil, which is called geophagia. Otherwise, you can also infect about accidental ingestion of dirt or soil and poorly sanitized hands. Humans can also get infected by contaminated plants, vegetables, for example, also herbs and fruits, which are not cooked, meaning raw consumed. And the last infection route is the route via larvae in peritenic hosts. This is due to infection of, due to consumption of raw or undercooked meat. Risk factors for toxoporosis are various. First of all, climatic conditions play an important role in egg survival, and it has been shown that subtropical conditions, meaning more than 80 degrees, provide an ideal environment for development of the infective larvae within the eggshell. Furthermore, humidity is an important factor because otherwise eggs would dry and die. These two factors, meaning subtropical conditions and high precipitation, lead to the fact that we commonly found toxoporosis more frequently at lower altitudes where temperatures range within 0 to 20 degrees most of the time of the years. Furthermore, environmental contamination with these eggs play an important role, and last but not least, also the socioeconomic status, meaning the environmental hygiene, sanitary behaviors, and also the given hygienic standards of such a population or community. Furthermore, it has been shown that age is a risk factor. It has to be said that, especially in children, clinical disease is often more severe than in adults. Nevertheless, adults are more often seropositive than children, which is due to a cumulative effect. And also pet ownership, or at least contact with animals, also including farmers, have a higher risk of being infected with this parasite. Overall, we can say that a heavily contaminated environment coupled with warm climatic conditions and poor hygienic standards provide ideal transmission possibilities. Now I want to introduce you to the community we have investigated. These are the Weber people, which are an indigenous tribe in the north of Colombia, which you can see it here in this maps. There is where these people live. They live in a reserve in the so-called Sierra Nevada de Santa Marta. Today, this community counts about 10,000 people, and they live a secured life. On one hand, due to cultural aspects, but they also have been historically victims of abuses, threats, and attacks by various other groups. And also today, they are still a neglected community in Colombia, and they also face the forced displacement by e.g. drug lords who use their land to cultivate certain illegal plants such as coca. The indigenous tribe of the Viva has a strong spiritual link to nature, and they live a simple community-driven life. Here on this picture you can see a typical home of a Viva family, and in these communities companion and farm animals have unrestricted access. You can see this on the slides here the dog and the pig, and here are the people eating. And the Viva people also have lacking access to electricity or clean drinking water and sanitary installations. The Viva have yucca or manioc as their primary food source, but they complement this with meat, fish, and some vegetables. We wanted to know if the Weber people particularly suffer from toxoporosis because all the factors for ideal transmission possibilities of these parasites are given in such communities. And what we did is that we sampled 483 inhabitants from 4 different villages of the Weber. These villages are Tetsumake, Chirua, Ashintukwa, and Sininke. And then we analyzed the serum samples with a Toxocara canis ELISA, which was commercially available, and results were analyzed by generalized linear models. What we found was a total Toxocara seroprevalence of nearly 80%, which was quite high. Borderline results we observed in 3.3% of the serum samples, and a good 70% were negative. For your comparison, Toxocara seroprevalence in Colombia as a whole is only 47%, and here you can see that we really have a high seroprevalence in this neglected Viva people. We also compared the different Viva communities and found that seroprevalence ranged from 75% in the village of Tetsumaque up to 90% in Seminke. We found that the two villages Tetsumaque and Tashintukwa, which are located at a lower altitude at about 700 and 750 meters above sea level, have generally lower Toxocara seroprevalences than Seminke and Chirua, and Tetsumaque and Ashintukwa are, besides their low altitude, also characterized by a tropical dry forest with a dry season of up to 6 months. And as you already heard, Toxocara X don't like a dry environment, and this might be the reason why we have a lower seroprevalence in these two villages compared to Seminke and Chirua, which are at a higher altitude and have either a tropical dry forest with an humidity of about 83% or a subtropical and humid forest environment, and this meaning increased soil moisture providing better survival conditions for Toxocara X. When we looked at the different age groups, we found that we had a clear increase from children displaying only about 67% seropositivity over the adolescents to the adults with 88% seropositivity, and this result was not surprising because adult or more old people are more and more exposed to Toxocara X, and so we have cumulative effects with increasing age. By contrast, gender had no effect. We did not see any significant differences between males and females. Yeah, when looking at the high prevalence of nearly 80% of Toxocara infected people, counteractive interventions are necessary, and I think one point which could be more or less easily be done is improve the water quality and the sanitation. Furthermore, people need to be educated that proper washing of their vegetables and avoiding ingestion of raw undercooked meat is important to prevent toxocorosis. But first of all, people need to be aware of toxocorosis, and this is why educational programs are needed in these neglected communities. Another approach would be the regular deworming of dogs and cats, for example, monthly treatments against worms, and such treatments or regular deworming programs would also be beneficial in the terms of the One Health concept. Another very effective measure to prevent toxocorosis is to dispose dog head feces containing these eggs. However, this is difficult to implement in the Viva communities because due to cultural aspects, the Viva don't touch feces, so they would never remove the feces of dogs and cats. And it's also quite impossible to prevent environmental contamination by wild carnivores. To sum up, as expected, we found a high seroprevalence in all examined Viva communities due to the ideal transmission possibilities for Toxocara X. We also found differences between the 4 villages, which can be explained by the altitude as well as the humidity of the environmental conditions, and we found the highest zero prevalence in the adult Weber people. As I already told, counteractions and preventions are urgently needed to protect also these neglected people from such zoonotic infections. Yes, I'm at the end of my talk, and I thank you for your attention, and I'm happy to take questions if there are any. Thank you.
All right, Professor Strube, thank you very much for your presentation. Are there any questions from the audience? Okay. If not, we're going to move with Professor Kietzmann, Professor Manfred Kietzmann. He's a colleague from the Pharmacology and Toxicology Department, and he's an emeritus professor. And before we attend with Professor Kietzmann, I'd like to make a little announcement that we've skipped our schedule. We've behind our schedule, unfortunately, and therefore we're not going to give a little coffee break. So after Professor Kietzmann, with 2 minutes, we will be starting with this session 2. So Professor Kietzmann, hello.
Hello together. I'm very happy that you organized this day together, Begüm, you and Bernd Lepenies in Hanover. I think it's a great thing. I remember about many things. When I was introduced at the veterinary school in Hanover as the Erasmus coordinator and later on as the head of the Office for Foreign Affairs, we had many contacts to Turkey, and guests from Turkey, especially Professor Bogo, who was mentioned some presentations earlier, asked me that we should accept 4 students from Ankara, more than the 2 which were in the contract. And this was a large discussion, and this discussion was decided lastly at one of the meetings in Hanover, drinking a lot of raki, and then we decided 4 students from Ankara will be guests in Hanover. And after this, I was sometimes a guest in Ankara, and the main impression I had in Ankara is the significant amount, the great amount of hospitality. You are expected. I was carried first time with a bus, with a transporter from the TO, which was used in Ankara for a long time. Now there's another transporter. We were going to the hotel, and thereafter the next point was coming, that is the Turkish meal. It was great, and every time it was great, and it's the only time in my life that we had after, I don't know, really 2 or 3 or 4 days that I must say, I cannot eat anymore. I'm so, it was such a pleasant food every time, and everything was very impressive. That is one of the points which is in my thinking. Other points are in my thinking which should not be presented here. I say one word which is understood by Begüm and some colleagues in Turkey, that is the word toilet, but I speak not about this wonderful thing. We had some colleagues at the veterinary school in Hanover. I will say 2 names that are Ludwig Haas, for example, who left us absolutely too early, and also Professor Salman, who were very active in the part from Hanover, the connection, and many persons in Turkey were also active. Then I remember that we had a meeting about pharmacokinetics over one day. There were many scientific and social interactions, and this day is a sign that it is possible. We have Corona and any problems, but we have the possibility in the digital way to hold on the contacts, and I'm very happy about this, and the large number of participants and presentators. So I think we will have good view on the future, and I hope that this will be proceeded in the near future. And I repeat many thanks to Begüm and also to Bernd to organize this, and I hope that we have a good future together. Thank you very much.
Professor Kietzmann, thank you for joining today, and thank you for your kind words. It's a great pleasure to see you back again. Okay, so before we start with the session 2, we will have a quick break. We will start at 12:45, and which means 10:45 in German time. Okay, so see you in 5 minutes.